Okay, so thanks. It was really illuminating so far to hear history about climate science, hear a little bit about the cave conservation. And what I would like to do now is focus a little bit more specifically on oxygen isotopes and what they're recording in speleothems. So this is really an introductory talk. There's a lot more details that I won't have time to go over, but it's going to focus on how do we interpret the samples? How do we recognize the well-behaved samples, the good samples, and how can we use that to infer something about paleoclimate? So my focus is paleoclimatology. And for that reason, when I'm working with speleothems, I'm looking for the stalagmites that have a strong climatic signal rather than a signal that might be complicated through cave processes. So in order to understand how that might work, we really need to go back to the beginning of looking at oxygen isotopes in the hydrologic cycle, and finally how they might be recorded into the speleothems themselves. Okay, so I'm going to start the talk here.
Matt, you're not in presenter mode, so if you want to just switch to.
Is it there now?
Yes, that's perfect.
Okay, so this is a photograph of Juan Pablo Bernal in Mexico looking at a nice stalagmite in the cave. This is not one that we worked on because it does not pass the photo test, but it certainly has a lot of the characteristics that we would be looking at, for eventually sampling and then measuring on the stable isotope mass spectrometer. So really, this is a summary of several reviews. You've probably seen many of these. The paper by Ian and collaborators in 2006 in Earth Science Reviews. I had a review paper in 2009, Ian and Baker's Speleothem Science book. And then this is a book that I recommend a lot for understanding isotopes in the hydrologic cycle. Environmental Isotopes in Hydrogeology by Ian Clark and Peter Fritz. I believe it's out of print now, but you can still find copies. And then finally, Zach Sharp's book on Principles of Stable Isotope Geochemistry. Second edition is available for free with PDFs online. You just need to look for it, do a search, you will be able to find it. So really, this is looking at the primary causes of oxygen isotopes in the climate system. Now, we're looking at speleothems, of course, because we're interested in reconstructing something about paleoclimates or paleoenvironments, or even the cave processes themselves. And so we can just ask this somewhat simple question then about why would we study speleothem oxygen isotopes? And the reason is relatively simple in principle, and that is well-behaved speleothems can preserve those variations in the oxygen isotopes of ancient drip water. And that ancient drip water has an oxygen isotopic signal, which is related to climatic processes. So when I use the term well-behaved, I'm mainly referring to speleothems that do not have lots of extra filters or extra complications on their oxygen isotopic signal. So in other words, the speleothems that are clearly recording something about paleoclimate variations. Okay, but to go back to the speleothems themselves, we have to remember that they're made out of calcium carbonate, at least the ones we're mostly concerned with in paleoclimatology. And if we go back to the carbonate equation, where you can take some calcium carbonate, add some water, CO2, it can dissolve to produce calcium ions and bicarbonate ions, or we can run this equation in reverse. Somebody's drawing on my screen, it seems. Okay, it's gone now. And the important thing is, if we look at the bicarbonate here, this has oxygen that's being equilibrated with the oxygen in the water. So the key understanding here is that the oxygen isotopes in that bicarbonate, the HCO3, assuming proper conditions, they're going to be equilibrated with the oxygen isotopic variations in the water. In that case, it's because the water is the largest reservoir. This is referring to the drip water, it's referring to the water in the epikarst, et cetera. So if there's enough time, there's going to be an equilibration of the oxygen isotopes between the water and then the bicarbonate. And then finally, this bicarbonate then precipitates back into the calcium carbonate. So we can potentially record those variations in the oxygen isotopes from the drip water in through the bicarbonate and back into the calcium carbonate. And that's why speleothems are very powerful, potentially paleoclimatic recorders with respect to oxygen isotopes. Okay, so that's the basic idea, but there's a lot that we're missing here. So we need to go back and look at what is controlling the oxygen isotopes in the hydrologic cycle, and finally, how are they incorporated into the cave environment. Now, this is a summary figure from the 2009 review paper that tries to break down the sequence of events in the hydrological cycle and all of the main processes that could potentially affect the oxygen isotopic values of speleothems. So if we start off to the right here with the ocean has a particular delta 18O value. When that evaporates, it produces vapor, which then has a particular oxygen isotopic value. Then when that water vapor condenses to form clouds and it starts to rain, that will change the oxygen isotopic value of the vapor and the rainfall. And then as that air mass evolves, as it transfers onto land, temperatures drop, might rise over mountains, et cetera. There might be recycling of moisture back into the atmosphere. And then finally, you get to this air mass that might snow or rain out onto a soil zone. And then from that soil zone, that water will make it into the cave until finally we end up with a speleothem here. Now, this particular diagram looks complicated because it is complicated to an extent, but if we break this down into individual pieces, it's not so complicated. But the point of this is that we can track the oxygen isotopic signal from the ocean into the air masses, into the precipitation, into the soil zone, into the epikarst, that zone above the caves, into the drips, and then finally into the speleothems. Okay, the key thing that we need to keep track of, though, are which of the processes are the most important for your particular study area, your particular cave. So what I am going to do through the talk is mostly focus on breaking this down into the different sections and talking about the main controls for these different locations. Okay, first, let's just get into some definitions and standards. When we talk about oxygen isotopes, or any stable isotopes for that matter, we're expressing them as per mil, or essentially parts per thousand. And usually we're talking about variations relative to some sort of standard. So delta 18O, the example of oxygen isotopes, is you measure the 18O to 16O value in your sample relative to some sort of standard, which is usually an international standard. It's always reported as relative to an international standard of either PDB or VSMO, and then we just take that value and multiply it by 1,000. So it's always really good to study stable isotope geochemistry a little bit with Zach Sharp's free textbook, or take a class to learn the basics of this. Okay, then we need to talk about how we talk about the stable isotopes. Because these are ratios, we just usually talk about values that are higher than the standard or lower than the standard, or higher or lower in relative sense. Sometimes you'll hear heavy values or light values, but I like to avoid using the terms enriched or depleted unless you're specifically talking about the heavy isotopes. So you could say, yes, they're enriched in 18O, but you could also say that they're enriched in 16O, which is depleted in 18O. So you get the idea that using enriched and depleted isn't as easy to understand. There's more sources of confusion. So I prefer just talking about heavier or lower, or sorry, higher or lower values. Okay, so the standards that we use for carbonates is the Vienna PD belemnite, or VPDB. When we're talking about it for waters, it's the Vienna Standard Mean Ocean Water, or VSMO. Now, the important thing to know is that both of these, because they're standards, are defined as 0 per mil. So really, we're just looking as it higher or lower than that standard. But the two are related, and they're essentially different by about 30 per mil. So we can relate the composition of VSMO to VPDB, or vice versa, with these two different equations here. So it is important to note that we have an offset. And a lot of some of the mistakes I see in manuscripts is not stating whether you're talking about VPDB or VSMO in your materials, and also confusing the delta values for VPDB with VSMO. So you always want to be very clear about what isotopic system you're talking about, because carbonates can be expressed in both. Okay, I'm going to have just a couple of short slides about stable isotope geochemistry and this term fractionation. Fractionation is what controls the isotope variations in the hydrologic cycle and in the speleothems. But it's actually a very simple process. And really what it is, it's just some process that changes the ratio of the heavy isotope versus the light isotope. So we can look at this, for example, changing the 18O to 16O ratio from the ocean water, let's say, to vapor that's produced from the ocean water, by using what's called the fractionation factor, this alpha here. And we can look at substances A and substance B. So we're just looking at, if you went, for example, from that ocean water to evaporating it into a vapor, you would have a fractionation factor here. And that can be calculated by just taking your delta value. Let's say A is the ocean, and then the delta value of the vapor, delta B, and then you just add them both to 1,000 and divide, and you come up with this alpha value. Now, finally, we, instead of using alpha, which is a somewhat cumbersome number because it's like 1.003 or some decimal point, we can express them in this terms of 10 to the third ln alpha. So you take the natural log of your alpha value and then multiply it by 1,000. And that puts us in numbers that are a little bit more easy to communicate. But it's also important for isotopic systems because they're related to variations in temperature. So I'm going to come back to this idea of 10 to the third ln alpha a little bit later in the talk, because it's important for assessing the behavior of equilibrium or quasi-equilibrium in stalagmites. Okay, there are a couple of different ways of measuring the stable isotopes. Typically with carbonates, we're either going to be using dual inlet or a continuous flow. But the basic idea behind a dual inlet mass spectrometry is you take your carbonate sample, dissolve it in an acid, and that acid will produce CO2 vapor and water. You separate out the water by freezing it, and then you essentially are purifying the carbon dioxide, and that carbon dioxide can then be transferred into the mass spectrometer. It goes into the ion source, which is a series of electronics, which ionizes it and then creates the stream that allows your mass spectrometer to actually analyze these samples. So this would be the type of system where if you have a Kiel device from the thermo systems for measuring carbonates. Another method is where we use continuous flow mass spectrometry. And what this does is similar. You take your carbonate powder, you dissolve it with acid. Again, you purify the CO2, but instead of transferring that gas to your mass spectrometer under a pressure gradient, you entrain it into a flow of ultra high purity helium. So that flow of helium will carry that gas into your mass spectrometer, and then you can measure your 18O signal versus your 16O signal in there. This is more typical with a gas bench type of approach. And nowadays, both of these approaches give similar precisions. They're both really good for doing speleothem analyses. So whatever works best for your lab is definitely fine. Now for waters, we have new technology, I say new within the last decade or so, laser absorption mass spectrometry. And this allows us to measure the oxygen isotopic ratios in water samples much more easily than mass spectrometry. At least that's what they tell me. So you can do lots of samples on one of these laser absorption mass spectrometers for waters, and then do your carbonates on the mass spectrometer. Okay, so those are really the three main ways that we get the oxygen isotope analyses of the materials that are relevant to speleothem work. Okay, so let's start going back to this diagram and talking about the main controls of the oxygen isotopes in different parts of the system. And let's start with the ocean. That's a good starting point in the hydrologic cycle because it's the largest reservoir of water. So we have to start off with knowing what the oxygen isotopic composition of the ocean is. Now you might say, well, we already know that it's 0 per mil, but the reality is that's not totally correct. On average, it's around 0 per mil. But it varies significantly from location to location on Earth as a function of things like evaporation, freshwater runoff from rivers, and on ice age timescales, it's varying as a function of things like ice volume. Okay, so let's just take a look at a map. This shows an estimation of the delta 18O value of ocean water based on measurements of surface ocean. And what you can see is that the delta 18O values can be significantly higher in zones of subtropical evaporation and significantly lower in the high latitudes where there's lots of runoff from rivers, for example. So it's not easy for us just to assume a value of 0 per mil, because in some locations it might be 1 per mil or even up to 2 per mil, for example, in the Eastern Mediterranean. Or if you're working in high latitude samples where there's lots of river runoff, the values could be significantly lower. So the basic idea is that when you have evaporation over the ocean, the 16O is preferentially evaporated into the vapor. And that leaves a concentration of 18O in the ocean water, so your values get higher. The delta 18O values are higher in the ocean water. But that also means the delta 18O values are lower in the vapor. And that's the vapor that's then transported on the land and falls out as rain and snow and goes into rivers. And then those rivers then have lower delta 18O values than the ocean. And then when those rivers drain off into the ocean, they can actually lower the delta 18O value of the ocean itself. So salinity and runoff are then correlated in this sense. They both are related to the delta 18O and the presence or absence of rivers and how much evaporation there is. Now on longer timescales, ice age timescales, we have changes in the amount of ice volume on land. And ice essentially represents stored ocean water that's been transported through the hydrologic cycle with very low delta 18O values. So when you get a buildup of a buildup of ice on land during the ice ages, that causes the ocean water delta 18O values to increase because you've removed preferentially a lot more 16O and stored it as a bank account on land. Okay, so that becomes important when we're starting to look at quaternary paleoclimate, because we know that the oxygen isotopic value of the ocean is changing over time. And one way we can look at estimating how big these variations are is looking in marine sediment cores. This is a marine sediment core on which they've estimated sea surface temperature in this top plot from the magnesium calcium ratios, and then measure the delta 18O values in the calcite. If you know the two of those, you can reconstruct the delta 18O value of the water. So yes, today, delta 18O value of the water is around 0 per mil VSMO, but we can see on the glacial, interglacial timescales, the last glacial maximum is about 1.2 per mil heavier than it is today. This is an inverted axis, so heavier is towards the bottom here. So roughly there's a change of about 1.2 per mil. Now, this could, in theory, be recorded in the speleothems. If there are no other changes in the hydrologic cycle, et cetera, you could expect to see a 1.2 per mil change in the speleothem if it grew over these same time intervals. Okay, now, then that ocean water is evaporating, going into the atmosphere. And this is where we start to look at the hydrologic cycle and oxygen isotopic values of it there. So here we have a lot more processes that are potentially happening, but which process is dominant depends on where you are at on Earth. So if you're working on a tropical island, let's say the Bahamas or the Western Pacific Ocean, it's a typically a more simple situation, because where what you have is you have a formation of vapor. That vapor then heats up and it rises, and then as it starts to rise, it cools off, produces condensation, the rain starts to fall out, and you have primarily a signal related to how much rain is falling out of that air mass. When we get to more continental locations, it becomes a little bit more complicated, because those air masses are also evolving. They're becoming colder, they're mixing with other air masses, they're raining out as they go. And every time you remove condensate from an air mass, it will end up changing the oxygen isotopic composition. Okay. And typically what you would see is you start off with warm temperatures during evaporation at the ocean. In the tropics, those air masses start to cool down as they move farther inland. And as they do, they're going to progressively be raining out more and more liquid or solid, the condensate, and that's changing and evolving the oxygen isotopic composition to essentially remove the heavier isotopes. You get lower and lower delta 18O values as you go from the tropics inland. Okay, so where your cave is relative to these different locations matters for which of these so-called isotopic effects are most important. Now, if we go back to the principles of stable isotope geochemistry, though, temperature controls basically everything in one way or another. And it does that through two main processes. One is just a direct temperature control, which is influencing the fractionation between liquid and vapor. So if you look at the y-axis on this graph, it's shown as the 10 to the third ln alpha. So it's the fractionation between the liquid and the vapor. And what we can see is that fractionation, the liquid is about 12 per mil heavier than the vapor at 0 degrees Celsius. And then that fractionation progressively becomes less and less. And of course, this is a high temperature experiment, 350 degrees Celsius. So we're not dealing with the hydrologic cycle on the right side of this graph. But what you can see is the fractionation gets lower and lower as the temperature increases, right? So that's one of the first aspects of how temperature is controlling the oxygen isotopic values of the hydrologic cycle. Now, it's also, we can calculate this. So we can calculate that fractionation between the liquid and vapor based on the temperature of our cave site, or based on the temperature of the ocean that's feeding our cave site, to give us some idea of what we would expect the vapor to be. Now, this is an indication of how the temperature is affecting the fractionation, but the next thing that happens is that when it starts to rain or snow, that condensate preferentially removes the heavier isotopes. And so the heavier isotopes will have somewhat higher values, and they'll also be removing those heavier isotopes from the air mass. So what this results in is an evolution of the oxygen isotopic value of an air mass as it cools. So we can take this situation of, let's say we start at the tropics where the temperature is high. If you look on the bottom left, temperature is high. And then as the temperature of that air mass starts to drop, we condense out more and more moisture out of that air mass. And this process is called Rayleigh distillation. So essentially what's happening is we're taking that air mass and we're squeezing out the condensate as it cools off. And as we do that, 18O is preferentially removed from the air mass as rain or snow. And that means that the remaining air mass has less of that 18O. So it gets progressively more negative over time. So this is a graph that is in the Clark and Fritz textbook, but I recommend everybody who's interested in oxygen isotopes to reproduce this yourselves by using the equations. So we're not going to go through the equations down here in detail, but you can do this yourself. So you can make your own graph that shows this evolution, and when you do that, you'll have a much better understanding of oxygen isotopes in the hydrologic cycle. So this is one of the assignments we actually do in my stable isotope class to help learn how this works. And if you haven't done it, I suggest you try it. So what this means, if we look at this dotted line, this is the delta 18O value of the vapor on the right-hand scale here. Maybe you start off at around minus 15 per mil delta 18O. As that air mass cools, the vapor is going to get more negative as it goes, at lighter and lighter values, because you're preferentially removing the 18O. Now, the liquid, in this case, has higher delta 18O values because it's preferentially incorporating the 18O. But as that air mass experiences progressive condensation, its value will also decrease. But they're separated by this value, the 10 to the third ln alpha value. Okay, so we could see that this is a decrease in the delta 18O values of the rainfall. And then the same would happen with snow as well, too, as you get to cooler and cooler temperatures. Now, these processes combine, the direct temperature effect on the fractionation and this Rayleigh distillation, produce lots of isotope effects that give us this global distribution of delta 18O. The point that I want to emphasize here is any process that cools an air mass to produce precipitation will cause a change in the delta 18O values of the vapor and of the condensate. So this could be related directly just to a temperature decrease. The temperature decrease in the air mass can also cause an amount of rain to fall out, a continental effect as the temperature decreases as you go inland, as the air mass rises up and over a mountain range, which is cool, etc. So it's important for us to understand where your cave site is relative to these broader patterns of stable isotope distributions. And you can find this information from oxygen isotopes and precipitation calculator. But if you do take one of these global estimates, you need to be very careful with that. It's better to use local data if it exists, or to test with local data against that, because sometimes these values can be very wrong in areas with more complicated isotope hydrology. Okay, so now let's talk about the meteoric waterline, because this is relevant for interpreting speleothems. This is a combination of both oxygen isotopes and deuterium isotopes, and we find that there's a nice relationship called the global meteoric waterline, where the hydrogen isotope value is just 8 times the oxygen isotope value plus 10. Where this meteoric waterline becomes important is when we look at things like the evaporation slopes, because that tells us if there's potentially been evaporation in our samples, in the epikarst, etc. Okay, so this is an indication where you have samples that plot to the lower right of this, that there's evaporation. And this is potentially important because if you're selecting a site to work in a cave and you measure the drip waters and they're consistently plotting on this evaporation slope, that means that they're not giving you a pristine oxygen isotopic signal. And what you might be seeing in a speleothem there might have to do more with evaporation in the soil zone or in the epikarst than actually in the climate system. Okay, so we do need to measure drip water values and then compare them to the meteoric waterline. On a global scale, when we look at the oxygen isotopic value at a particular location in space, we do see that there's a strong correlation with temperature. And this is particularly true when we look at the full range of temperatures from, let's say, Antarctica, minus 50 degrees Celsius into maybe the subtropics, 10 to 15 degrees Celsius, but it starts to break down in the much warmer areas in the tropics. Okay, so this is called the temperature effect. If you're working in a mid-latitude location, this might be a dominant control on your cave site. So it works best in the mid to the high latitudes. But it's also partly related to seasonal cycles about where the rainfall is coming from. In North America, for example, you might have the Gulf of Mexico versus the Western or versus the Pacific Ocean. So those two different air masses might be giving you different values, but they also are varying seasonally. So here's just some data that shows that for one of my field areas here in Nevada. What this plots on the left is winter temperature versus the oxygen isotopes of precipitation. So you can see as winter temperature increases, the oxygen isotope values also increase. So here in the Great Basin of Nevada, we would expect to at least have some control on temperature on interpreting our speleothem oxygen isotope values. And we can break this down into more detail by looking at where those air masses are coming from, what the moisture sources are. And usually the air masses that are coming from the high latitudes, in this case, they're in the Pacific Ocean, have significantly lower oxygen isotope values. And then the more tropical sources that you see during the summer have higher values. So this is the type of work that you would want to do at every field site where you're collecting your samples. I don't like to rely on oxygen isotope precipitation calculators. I like to do these analyses myself to make sure that we understand what is going on from these. So this is kind of a recommendation, is try to find this data at your own cave site, or if you don't have it, start collecting it. Just this last point here, the relationship that matters between oxygen isotopes and these different climate parameters is the one that's relevant to your field site. So you need to verify that. We also note that there's a strong seasonality effect, and this seasonality effect can change over time. So this plots monthly precipitation delta 18O for different locations. At the high latitudes, you can see that the summers have significantly higher delta 18O, the winters are lower. Again, you want to verify this at your own field area. So on the right, these are histograms from precipitation in the Las Vegas Valley, which I collect at my house and on the top of the roof of my building here. And you can see that the oxygen isotopic values of winter are significantly more negative than those during the summer. So create this for your own cave sites, these types of plots, to help you understand what the dominant controls are. Okay, I'm going to talk a little bit about the amount effect. The amount effect is the observation that in some tropical locations, the delta 18O values of the rainfall are related to the amount of rainfall. And the basic idea is you take one of those air masses that evaporates over the ocean, you heat it up during convective processes, and as it rises, it cools, and then rain falls out. And the more it rains, the more the 18O is removed from the air mass, preferentially due to the Rayleigh distillation, and therefore the lower the delta 18O values. So you see there should be an inverse correlation between rainfall amount and oxygen isotopic values in this simple sort of setting. And the data in the graph here is related to, let's see, this is Panama. So the green circles are monthly values for delta 18O related to precipitation amount. So you see a seasonal cycle. And then the blue squares are the annual values. So you see this on a year-to-year basis, but it's a little bit weaker. So you could expect to see this more in tropical locations where you have this simple relationship between forming an air mass and rain out. Now, what we've learned, though, in really the last 10 or 15 years or so, is that when we talk about the amount effect, we have to be very careful about what we're referring to. It was originally described at a location, say, when it rains more at my field area, the values are lower. But what we're starting to find is that what's more important is rainfall amount on a regional basis. And so it could be that if you're working just on a small island, you might have a very simple rainfall amount that's related to local rainfall and oxygen isotope values. However, if you're working in other locations, it's significantly more complicated. And that's because how much rain falls out of an air mass upstream of your location might be more important than how much rain falls at your particular site. So we've now kind of changed our understanding of the amount effect to infer its changes in the regional amount of rain, not just at your particular field area. And this is important because if you measure a tropical speleothem, you might see what you think is a rainfall amount, but it might not reflect your field area. It might reflect what happened to those air masses upstream of them. So I really like this paper from the Australian monsoon where it made this this observation very strongly. It says the strong correlation between rainfall isotopic variation is really related to integrated rainfall history of air masses prior to arriving at the measurement location. Okay, so that's something to keep in mind. Again, you can study this by doing the isotope climatology of your own sites. We also see that there's significant source effects. Changes where the air masses are coming from might have significantly different oxygen isotopic values because they have unique histories. Because the integrated upstream rainfall can vary from one source to another. We can back calculate that using back trajectories. High split is commonly used. There's other ways of doing it as well too. And so you can just measure this in precipitation over a year, a couple of years or so, and see what the relationship is, finding out where the air masses are coming from and what their unique oxygen isotope signal is. When you do that, you'll find that there are often unique sources that have unique oxygen isotope values. So again, just going back to Nevada here, just for example, when we have air masses coming from the polar regions, we have significantly lower delta 18O values versus the tropical regions, which are significantly higher. Then we have some intermediate values in between. So again, work this out for your field area. Now, what do you do if there's no data from your field area? Well, you can collect it yourself over time. That takes many years. If you're doing a master's thesis or a dissertation, sometimes you don't have time to do that. One of the things you can do is measure over space and then use those isotopic variations over space and correlate them to climatic parameters, right? So this is just one example of, in this case, of using surface waters to try to look at the oxygen isotope variations. This case being the South Island of New Zealand, and then you can come up with maybe some sort of model, and you can model the oxygen isotopic values geographically based on the spatial sampling. Okay, so in this case, it gives you actually predictions of what the delta 18O values should be in different locations. So if you're working on a speleothem up in the North Island, or excuse me, the north of the South Island of New Zealand, here it would have a very different value than if you're in somewhere around central Otago in this case. So I find these types of isoscapes very useful for understanding as well the climatic controls on the precipitation that's coming in. Just some examples from here. We already talked about this rainout effect, the so-called Rayleigh distillation. Here we have a transect from these water samples across the South Island of New Zealand. These are the delta 18O values. They decrease as we go across the island to get their minimum values sort of in the middle, and then they increase back again. So basically what we have is we have two air masses coming in, and towards the coast you have high values, and in the most interior continental locations, it's really only a single air mass coming in from the Tasman Sea, so that has the greatest amount of Rayleigh distillation. Okay, so again, this is the local data that helps you understand your potential field area. Okay, so finally, you've done all of this work. You've got these plots, back trajectory analyses of the isotopes and precipitation. Then you need to look at your cave site. So what we have in this case is the rain or the snow falls onto the soil zone where it can be mixed together. It can be stored over time. That water then starts to infiltrate into the epikarst. It can follow fracture conduits. It can follow diffuse flow until it finally ends up as a drip inside of the cave. Okay, so here we have the atmosphere, and then we have the filter that's related to the epikarst and related to the soil zone before it even makes it into the cave to be turned into oxygen isotopic variations on the speleothem. So I like to use this term of isotopically effective infiltration, and that is it's the water that makes it into the cave. And it's that water that makes it into the cave that's precipitating calcium carbonate, because not all water that makes it into the cave will actually be saturated with calcium carbonate. In fact, we know that where you see these drip dissolution holes in speleothems, that you can have infiltration that's unsaturated, and it can actually cause your speleothems to be dissolved away. So this is where it gets important to go into the cave and start measuring the drip water samples and making observations of the speleothems. If you see caves with lots of these dissolutional holes on them, you need to be very careful about that cave. Because they might have lots of drips that are undersaturated, which means your speleothems might also be affected by dissolution over time. Now, I like to say that each cave is unique. Sometimes each drip is unique, and you don't know that until you start analyzing them. And that's why we need to be doing this monitoring data to the best of our ability inside of the caves to understand what's controlling those oxygen isotope values. So a couple of nice papers have looked at, how climate and this processes in the epikarst and in the caves can have influences on the oxygen isotopic values of the drip waters and into the speleothems. Here's just one example. Take all of that rainfall that falls over a cave. What happens with the isotopic signal is it tends to smooth it because of mixing of those waters. And it also dampens the signal, and it delays the signal because it takes time for that water to move through the epikarst. So the plot on the right is a histogram where the bars are showing the delta 18O values of precipitation above the cave site. And the very narrow dark colors in the middle show the delta 18O values of the drip waters. So you have a much more restricted value because of smoothing and dampening of that signal. And in some cases, this also can be related to the bias of the cave. So maybe the caves are only receiving infiltration associated with perhaps winter precipitation or the most heavy rainfall events, but the lighter events or maybe summer precipitation isn't able to infiltrate into the cave because it's being used by plants. Okay, so we need to understand those epikarst processes. Okay, finally, let's get into speleothems, which is kind of the point of this, and other people are going to talk about this in perhaps a little bit more detail, but the assumption is that we take all of these different processes and it ends up with a drip water value, and that drip water value will then be incorporated into the speleothem with some sort of offset. But we really need to test this kind of more quantitatively and sometimes qualitatively. And I like to use kind of two tests. One is you look for whether the speleothem is forming in approximate equilibrium. And then the next is with your speleothems, will the records replicate between different speleothems? So I only have a few minutes, so there's enough time for Q&A after. So I want to go somewhat quickly through this. But the first is the equilibrium test. We can estimate what the delta 18O value of a speleothem should be based on knowing the cave temperature and the drip water delta 18O values. Okay, there's some different equations for this. My current favorite equation is this Baron et al. 2019, which it calculates the 10 to the third ln alpha between calcite and water And really the only value here is temperature and the delta 18O value of the water. So what you can do is calculate that value for your particular speleothem in an equilibrium test. And what this shows is the fractionation versus the temperature. And there's multiple curves here. Each curve represents a different estimation of fractionation. And you can calculate that for your particular speleothem. And if your value falls close to one of these curves, you can have more confidence in it. But if you fall way out, let's say on one of these areas here, in what I like to call the zone of danger, it's suggesting that your speleothem is not forming in equilibrium or anywhere close to equilibrium. And you need to be very careful with samples that plot there. So there's more details in some of my papers, which I can send, or you can just calculate this yourself based on the fractionation factors. The final, which is really more powerful in my opinion, is what we call the replication test. And this is once you have the speleothems and you have the age models, do the oxygen isotopic values replicate between different stalagmites? So to do this, you need to have more than one stalagmite, which is always complicated because it's extra work, cave conservation issues, etc. But if we can replicate them, we have much more confidence. So here's just an example that I put together. This is data from Borneo. Here's, let's say, a benchmark speleothem, multiple different speleothems kind of overlap. And we see that they're not perfectly overlapping, but they do a pretty good job at the overall signal. And so what these authors did is they applied corrections to kind of realign the different speleothems to produce this replicated record, which goes back 100,000 years. And now we can have much more confidence that what we're seeing is a true climatic signal. Here's a bad cave. Bad cave. The records do not replicate at all. When you find a cave that looks like this, you can try to figure out why, or you can just find a new cave. My approach is to find a new cave, get away from the bad cave so that we can actually get something about the paleoclimate. Okay, so finally, which isotope effect is important at your site? Do the literature review or do it yourself if you can't. Attempt the calibrations, whatever means possible. Calibrate the precipitation, the drip water, the speleothems, etc. And a really nice study is this by Lachniet Hernandez et al. That looked at this at a cave site in the Yucatan Peninsula. Okay, so I think I'll stop there so we have a little bit of time for Q&A.